My talk is about efforts of my lab at the Chumakov Scientific Center on the search of the new antiviral compounds. COVID pandemic does not leave anybody outside of it, and all of us now know that antiviral drug discovery is a really important problem of the contemporary medicinal chemistry. And the scope of the problem is really large, really great, because natural diversity of viruses that could go to the human population is really wide and large. For example, International Committee on Taxonomy of Viruses now has more than 10,000 viral species defined according to their characterization by genetic methods, and at least 200 of them are known to infect humans, and at least 3 to 4 new human-infecting species are still being discovered every year. So by now it could be estimated as at least 300 viral species pathogenic for humans. Viruses as a whole are very abundant objects, and they are estimated to represent, you see, 10 to power 31 particles on Earth and 10 to power 13 particles per human body. And it shows that you cannot even know how many different particles are there and how many different pathogenic outbreaks could arise from them. And finally, the Global Virome Project tries to prevent the future pandemics, and they estimate that at least 500,000 undiscovered annual viruses could be capable of transmission to people. It's really easy to understand because almost every animal species has its own viruses, and any contact could be potentially harmful. And virus species-specific viruses are not unique but really abundant. On the other hand, we have a diversity of antiviral drugs, which is really rather low. We have only about 100 different antiviral drugs, and 120 if we include antibodies, biologicals, combinations, and drugs approved at small jurisdictions, not like FDA and not even like Russia, but much smaller countries. These drugs mostly target only 11 different viruses, about 15 different diseases caused by these viruses. So it is understandable that we need to explore this chemical space to find more and more potential antiviral drugs, which could be helpful against new potential outbreaks. Most common classes of antiviral drugs include nucleoside analogs and peptide mimetics. It is clear, and most antiviral drugs have lots of clinical limitations. For example, they often have an inconvenient spectrum of side effects, or could not be used systemically, and could be used only by injection or liniments. Finally, due to similarities between the antiviral drugs, we have a lot of repurposing options, and we are trying to discover, in the perfect case, the broad spectrum antiviral drugs. You could also think that vaccination is a common procedure that could be used to prevent viral diseases, but you see there is a lot of different problems, because vaccines are biologicals, so they require cold chain to be transported. They do not usually provide opportunities for treatment after onset or after contact. There is a lot of anti-vaccine movement. There are issues with community immunity generation, and not all vaccines are completely efficient. They do not guarantee you against the disease. So it could not be easier in general to generate, to use vaccines instead of antivirals. Here you can see the probably newest antiviral drug that was approved, and you see that it definitely belongs to a very similar chemical series. You can just consider these molecules as brothers. The first one of them is a selective covalent inhibitor of HCV protease. Another one was developed during the first SARS-CoV outbreak, but never progressed to clinical studies because this outbreak was eliminated by itself, and there were no cases. But only because this drug existed, the nirmatrelvir could be disclosed after a year of COVID pandemic and approved after 1 year and a half. In our studies, we are trying to explore different complementary approaches to antiviral drug discovery: phenotypic screening and target-based screening, both in silico and in vitro. The same types of screening in silico and in vitro do provide a feedback for themselves, and you usually need to do both of them. But generally, every screen should be validated experimentally, and when you take a phenotypic screen in silico, you should validate it phenotypically in vitro, and then probably by target-based screening in vitro. If you take a target-based screening approach, you should validate it by target-based screening and by phenotypic screening. Also, you can validate your target-based screening in silico by phenotypic screening in vitro, but this situation is slightly less easy because you do not prove the mechanism in that way. And also, if you don't have a possibility to do a target-based screening in silico, you can validate a phenotypic screening in silico by target-based experiments in vitro, but still you will need to do some phenotypic screening. And this way is useful only if you want to keep with a certain target, taking compounds from phenotypic screening. And obviously, after the screening validation, you need to do in vivo assessment for development of drugs, and you need to validate your target and your mechanism. So additional studies are always required after the screening. To have a success with virtual screening, you need to curate your data, and in the data-driven world, we have only 20% of early discovered data of high confidence and large errors. And it means that we need to clean up the data and sort them out to make the search in the databases of phenotypic screening in vitro, in silico, more convenient. And if you take the most common database, Campbell, we can see that we can not even easily extract the data for a single virus. For example, if we take a hepatitis C virus, we have a huge amount of different genotypes, and we need to unify the data for all of them. And for other viruses, we have even the name variations that could be not similar to each other, and you could not just take a single search string to extract all the data. Thus, we have performed a taxonomy-based annotation of Campbell data using ICTV taxonomy as a guideline. As a result, we have constructed the database Viral Campbell, which uses a sophisticated data annotation procedure to taxonomy, and you can see that even the data extraction is very efficient if we take our approach in mind. And still, this data set which we extract from Campbell is very sparse, because for most compounds, the data are measured only against a few most common viruses. But still, this database is large enough to perform some data mining and data analysis. We have used approaches of chemical space in the first applications of Viral Campbell to real problems. And we started with a generative topographic mapping, which was a collaboration with Professor Warnack. In this approach, ECIDA fragment descriptors are used for the database description, and training set and screening library are projected onto the universal maps of Campbell. And then we select compounds with similar responsibility patterns to the good screening hits, to the active compounds. We have validated this study by purchasing 48 compounds tested for anti-tick-borne encephalitis virus activity, and we got about 50% hit rate. And 4 compounds were new chemotypes with micromolar level of activity. So this approach shows that it is definitely successful. Another approach, very similar, still chemical space analysis, but with the help of self-organizing maps. We have used data warrior implementation here. And here we first construct a map for the target library and then project the training set onto it. And then if we have a good neighborhood for active compounds, we take the molecules from this neighborhood and test it. In this approach, we have purchased almost 200 compounds and tested them against 2 viruses, 2 flaviviruses, TBV and yellow fever virus. And we have also a very good hit rate, rather lower, but what is more important, we have a few anti-yellow fever virus compounds, which are very rare in the medicinal chemistry practice, but yellow fever is one of the most common viral diseases in the tropical regions. Next approach is just a phenotypic screening in vitro of the privileged structures. It is very hard to develop a good model for nucleosides or for pair lengths, and it is easier just to take a series and screen it. And it was what we done, and we have found several very potent compounds in this series. Only a few examples are given here. For the heat compounds, we need to make some mechanistic studies. For example, in the case of this compound series, we performed an antiviral activity spectrum analysis against different strains of tick-borne encephalitis virus. And the most interesting findings here is the strain 256, which is identical to another strain upsetar by the target protein sequence, but all compounds are much less active against this strain. What is the reason? The reason is that this strain contains much more decoy particles. It means non-infectious particles, immature particles, and destroyed particles as quantified here. So the reason of the lower activity is that decoy particles take the antiviral and make and do not allow it to target infectious particles with the reasonable efficiency. In a clearer experiment, we have directly generated systems where a strain was mixed. We have prepared a mixture of mature particles, which are infectious, and immature particles. And in this case, we have shown that immature particles could bind the antiviral, and then EC50 becomes very large. So it also proves our hypothesis that these compounds interact with the envelope particles of the envelope proteins of the virus. Next approach is the target-based screening in silico. If we have a homology model, so we can dock compounds of interest into it and try to find something new. This is our very first study when there were no there were no tick-borne encephalitis virus inhibitors at all, and we have just performed a docking and phenotypic screening validation because we don't have a protein-based system for binding measurements. And you see that some of these compounds are still the most potent among the tick-borne encephalitis virus inhibitors. Another approach we have exploited with the SARS-CoV-2 main protease. When we first performed a GTM-based screening to make the docking a program much smaller and affordable, not to dock all the zinc, but to dock only the selected compounds which are similar to other coronavirus inhibitors. And you see that after taking a huge amount of compounds from zinc, we have docked only about a few hundred compounds. Then we purchased 10 of them, and 2 of them were active in vitro. And for more information, you could hear today my PhD student Anastasia Fomina, who will take about these studies in much more details. So we have validated our EMPRO inhibitors using thread-based assay. Oh, sorry, this picture is distorted. And you see that, well, they are not so good as typical specific inhibitors and covalent inhibitors, but this activity is definitely optimizable. And in general, we have an in vitro screening system which we could validate both with EMPRO-based assay and virus-based assay. And you see that in our screening, we have about of the set we tested, we have about 20 actives of 115 dozen. To summarize, in our studies, we have developed in vitro and in silico screening approaches that could give a specific attention to discovery of antivirals, and we validated these approaches using the experimental methods. And we can use these approaches to solve practically relevant problems in the antiviral drug discovery. We have discovered a few new antiviral chemotypes which are being studied in details with the hope to optimize them as possible drugs, or to study the mechanism of actions so they could be the analogs could be synthesized. And also we are doing our structural virology studies to try to find the most direct interaction data between the proteins and the small molecule antivirals. This work requires a huge amount of collaboration. This work requires different sources of funding, and I would like to thank all these people who participated and who donated in some way, especially the people who donated their compounds for us to study. And I would like to thank the organizers for the possibility to make this presentation. And finally, I would like to thank you for your attention. Thank you very much.
Thank you, Dr. Dimitri, for your nice presentation. So we have received one question in the chat box from Dimitri Shulga. What is phenotypic screening in Silico? Is it a sort of QSR?
Phenotypic screening in Silico is, well, all the types of phenotypic screening in Silico which do not rely on target specific effects. For example, if we have antiviral activity, which is a phenotypic characteristic, we can use it as an endpoint and predict it. So it is a phenotypic screening in Silico. So it doesn't have a target-based endpoint. Usually, this is machine learning, this is chemical space analysis, and this is some QSR approaches, but the most important is the endpoint which we are trying to predict. And usually we don't predict antiviral activity using docking because it is not relevant.
Okay, thank you. So there is no other question, but I can ask you a question. So what do you think about application of machine learning in antiviral drug discovery? So will that be a future avenue for getting more promising drug candidates?
Oh, sorry, I could not hear
application
the
of machine
part
learning,
of your question.
Application of machine learning in antiviral drug discovery.
The problem of machine learning regarding the antiviral drug discovery is data. Because as I showed, the data are very sparse. And in the current situation, in the most cases, it is possible only to predict an activity against a small, well-defined group of viruses for which we have a good training set. For example, HIV or SARS-CoV-2 or HCV or something. I'm sorry. There are also approaches which I did not discuss here, which are developed in the collaboration with Skoltech, the laboratory of Maxim Fedorov, Sergei Sosnin, and Ekaterina Sosnina, who tried to use the viral Campbell data and to apply the recommender system approach. You may know recommender system as the system of e-commerce, which helps you to buy something similar to you previously bought. Yeah, like you know, on Amazon you have some recommendations, or you have recommendations on Netflix. It uses the same idea of machine learning, but to predict compounds with a similar profile of activity. But in all cases, the database becomes too sparse, and we need to measure as many points as possible experimentally to make the predictions better.
May I also ask?
Sure.
Okay, you mentioned the restrictions in application of in silico methods for antiviral drug discovery. What do you comment regarding the contradictions in vitro data obtained in different assays?
I should have taken a very nice figure which shows sources of errors in vitro assays. If we take, I'm sorry, if we take target-based assay, we have a few points of variation, like the sequence of enzyme, the pH of the buffer, the quality of the substrate and I don't know, the quality of the DMSO. And if we take a cell-based assay, we take variation with the viral strain, we take the variation with the cell strain, we take the variation with everything in general, because even the ambient temperature could influence the results, despite most of the time the cells are sitting in the thermostat, but during the preparation they could be affected by additional problems. So the problem is that reproducibility is bad, but we don't have anything better. And well, I can only just show this picture again. We are trying to sort the garbage, but in many cases it remains still the rather garbage data, but some predictive ability could be inherited from them.
Okay, thank you.
So I can ask you another question that
Yes.
in view of anti-COVID drug development attempts, as you know that different categories of drugs are being tried for drug repurposing against different targets of COVID, and not only antiviral categories, but different other pharmacological category of drugs are being tried computationally for drug repurposing. So what do you think about this?
Our experience shows that computational drug repurposing is often helpful because drugs are very easy to buy, and you have start your experiments with something that should work and often works really. And we also have some positive results, which I don't think I should discuss right now, because it is a project in its course and it is not finished yet.
Okay.
So computational repurposing is good as all the ways of repurposing to find the first hits.
Okay,
And
thank
then
you very much.